There is a secret world of tiny animals living in and even creating the soil underneath our feet. It's a vital part of how the world works, and yet most people have no idea what's going on. They don't have any idea of the biodiversity down there. So let's dive into this secret world, see what we can understand about it, and see what it can tell us about life in general in the process. Paleontology is about understanding the history of life. Now, you can do this in various ways. Partly you can take some stones like this with little graptolite fossils on from 460 odd million years ago, and you can try to understand from looking at the rock itself, the environment they were living in, think about the filters that affect how likely something like that is to be buried in the first place, then to be preserved in a way that it will then be recognizable, to be uplifted through tectonic forces, and then finally discovered by a paleontologist. You can think about all those filters and then try to work backwards from what little is left to what there once was. Or you can look at the modern world and try to go backwards. Now, there are a lot of people out there who are dedicated naturalists who spend hour after hour just watching the birds or walking amongst the trees and just feeling the wonder of the sheer beauty of this incredible world of biodiversity that we live in. And many of these people feel that they understand nature. But nobody really understands nature. I certainly don't. Because there is so much of it. No matter how much you focus on, how much you try to expand your sphere of knowledge, there is still far more that you will never really come to understand. So, one of those areas which is sadly neglected and that hardly anyone has any clue what's going on is the soil. The fundamental basement of all of the rest of this life, the birds, the trees, the insects, everything depends on there being a source of nutrients and a substrate for everything else to grow in. And the soil is a vital part of our world. And it is full of biodiversity. I don't mean just microbes, bacteria, fungi. There's a huge range of things there. But it is also stuffed full of tiny little animals that most people just never meet. If you're digging in the garden, you might find earthworms and some centipedes and things like that, but they are the very rare large organisms that are within our notice. Because you see, humans are gigantic. We are absolutely enormous in comparison with most life. And so most things we don't pay any attention to because we don't see them. Our eyes are not designed to look for things within a centimetre of them so that we can actually see these tiny little mites, for example, running around in the soil, living out their lives in a way that we'll never experience. But in order to understand what is in a place, we do actually need to find ways of discovering that. As well as being a paleontologist, I'm also a bit of an entomologist. So I do bits of survey work where I go to some land and try to understand what is there, make a list of the species that we can find, their abundance, and try to work out a bit about their ecology, how it relates to the landscape, to the geology, to the wider ecosystem, and to how humans have affected it. And when you do this sort of work, you very quickly build up a different understanding of many aspects that are relevant to understanding the fossil record. So, for example, the concept of rarity in the modern compared with the fossil record is a totally different thing. And this is something I'm going to be diving into quite a lot more in some future videos because I think it fundamentally affects how we relate our ideas about living ecosystems into the fossil record and try to understand the extrapolation of them into the deep past. So this is a vital part of trying to understand the fossil record, is understanding how life actually works today. And I don't mean sort of theoretically, I mean the practice of it. I mean, going out, seeing what there is year after year until you build up an understanding of distribution patterns, abundance variations, seasonality, all these aspects that are not immediately obvious when you're looking at lumps of rock that preserve a single snapshot from an undefined moment. Now, most of the work that I do is based on macro photography. So I go out with a big camera and macro lens and extension tubes, and I can record most of the things I'm interested in purely from photographs. There are exceptions. There are things where you cannot identify a species without dissection or DNA analysis or something like that. And those I will take and perform the necessary duties on. But for the great majority of things that I do, I can study them purely from photographs in the natural habitat, which is great because it makes me feel a lot better. The problem is that not all of life can be studied in that way. So, for example, the things that live in soil is something I've been getting into over the last 5 years or so, the mites and springtails especially, and these are extremely small and they are very difficult to identify in most cases. So you have to have a different technique in which you basically extract them into something, in this case alcohol, which can be put into a slide microscope to look at minute details that you could never see when they were alive. I don't like doing this, I'll say this at the beginning, but then I do make up for it by, for example, never cutting the grass, and I don't drive. Anyone who does either of those things will be destroying far more little living creatures than I could ever hope to do in a lifetime of surveying. Hope not to do, you know what I mean. So with that caveat aside, I don't enjoy taking destructive samples at all, but if we want to know what is living in an environment, if we want to understand it, there has to be a small amount of that done, which at least is a sacrifice that allows us to then understand and potentially conserve habitats, rather than random destructiveness that most people do accidentally. So I'm fully aware that it's not ideal, but I do what I need to in order to help in the longer term, trying to understand what is living here and how. So there is a site that I'm working on at the moment, and thank you to my employer. You know who you are. And this is a wonderful little farm area in mid Wales, which I will say no more about for privacy reasons, but it has some very lovely features of it, mainly due to the modern management of it. And it includes a whole series of different fields, woodlands, and so on, which I can try to understand by first of all seeing what is living there. So I'm doing a series of samples where I go to different parts of this little farm and then take a little sample, analyze it for everything that's in there, piece it all together to try to work out what it's telling us about what that ecosystem is really like, how unusual it is, how it relates to the wider area, and how it relates to the local geology and the land use, how everything fits together, in other words. So you have to select your sample sites very carefully, because when you're taking a handful of soil and moss and you are then examining that minutely, so you want to make sure that's fairly representative of the wider area, or even better, actually tells you a maximized view of the diversity that's living there. So it's quite fun picking your sample site. When you're looking for a place to take a soil sample, it's not the easiest decision, because you're taking a tiny little bit of soil and moss and leaf litter to see what's living in it. But it takes so long to go through one sample, maybe a week or so if it's a good one, that, well, you can't really just sort of pick one at random. You've got to make some sort of choice as to where you're going to look. So some things you might consider is rotting wood, especially really old wood where it's decaying into the soil. There'll be lots of things living in there. Moss is a good one as well. that lovely little sort of green carpet that you get either on rotting wood or on the soil underneath it, or at the bases of tree trunks, for example. There's going to be all sorts of things living there. Moss is actually a really good habitat. But then, do you want wet or dry? There's a stream over there as well. Do we want the side of the stream, or do we want somewhere that does actually dry out sometimes? Because there will be different things living in every area. When you think in terms of birds or something like that, then this is a habitat. When you're thinking of mites and springtails, then there are 1,000 habitats here. And you've just got to pick the one you think looks most interesting. So I've decided to go for this one. We're at the base of a great big oak tree. Well, it's maybe a couple of 100 years old, not exactly an ancient one, but rather lovely. And at the base there is a thick swathe of moss covering the floor. There's really deep leaf litter here where it's all built up. And there's also very thick mossy ground and liverworts on these trunks as well. And fungus growing on the trunks, that's always good. Well, branches. So we've got decaying wood, lots of fungi, lots of mosses and other plants. Right underneath in there, I think, is going to be the best place to sample. And the process is pretty simple. I've got a little trowel and I will take some of this soil here, avoiding the big bits, and put it into a box. Very, very high-tech this. Trying to make sure I get some of the deeper parts of the leaf litter, as well as some of the moss. Take some of a little bit of the wood that's growing here with the moss attached to that, just to get a little bit more variety. There'll be things living there. And go in a little bit deeper to the subsurface. There we go, that's actually into the soil. And there we go. That is a sample. Ta-da! Once you've taken your samples and brought them back home again, to find out what's in them, you need a setup like this. For something like soil mesofauna, mites and springtails, unfortunately the only way to identify them is to pickle them. And that's why we have a Tulgren funnel. So what we've got here is the sample is in a sieve, the sieve is in a funnel, and the funnel feeds into a little pot of alcohol. Now you'll notice this is next to a radiator. It's a very high-tech setup. Most people have a heat lamp, but you have an array of heat lamps above each sample. But the basic premise is that you heat up the sample from above, slowly dry it out, and all these things that like the cool and damp conditions go downwards through the sieve and drop into the alcohol, where they can be identified at leisure. So that is what we've done here. And this is one sample I'm going to show you. So I'm just going to take this out carefully without shaking it, because then you get whole piles of mud going in, which makes it an absolute pain. And here we have our little vial with alcohol in there, 96% ethanol. The reason for that is that then we can actually use DNA barcoding on it. The ethanol doesn't degrade the genetics. So if there's anything particularly interesting, I can extract this, keep it in quite sort of clean conditions in a separate little vial of pure alcohol, and then we can do something with it. But that there is a sample full of everything that was living in this tiny sample of soil and moss. And within this, there might be rather a lot of different species. Do you think we should go and have a look? Yeah, go on then. So the first stage for looking at things is with the trusty old S8 APO, which is a nice binocular microscope. It goes to reasonably high magnification, but is mostly quite low powered. And we can do a quick sort of initial sort, we can see what's in there, and we can take it from there. So this is the sample, and this is a little dish that we have to put it into. So let's do that first of all. So you can sort of swirl it round to get everything as suspended as possible, and pour it into there without spilling it all. Now there's quite a lot still stuck in there, so we use the standard tool for this sort of job, which is like this. And anything left in there now is going to be bits of mud, basically, because that tends to not be picked up as much or as well as things like little springtails and so on. So I'll pop that in there. Just keeps it nice and handily upright. And we now have a little sample, which we can put under here and see what is in it. And here we have it. So immediately you can see there are quite a few springtails. These are entomobryomorphs. There's several major groups of springtails. 4 of them are, you're quite likely to get all 4 in one sample. And that is quite an interesting little one. We'll have a good look at this one in a bit. There's smaller ones there as well, probably the same species. The fact that there are multiple ones in here is actually still useful information. It's not wasted by duplication because I will be counting all the individuals that you get in these samples as well. And then when you're comparing different samples, different habitats, you can see how the ecosystems are different. And that's a vital part of understanding what these things are telling us. And, ooh, that's a nice one. That there is one of the orobatid mites. I'm going to have to change the contrast on this one. And this you can see is an extraordinary little creature. I know it's very dark, so you can't see much detail, but it's got its legs sticking out the front. And I'll have to look at that with better light to check which one it is. But it might be something like UZT's globulus, possibly, which is quite a common one. But I think that's the underside we're looking at there. We'll need to turn it over and have a look at the upper side. But that is an aphid, a little baby aphid. Okay, there are quite a few aphids that feed on roots and plant roots underground, and it's probably one of those, but that's not something I can identify easily. Certainly not a juvenile, or possibly even identify at all. Now we've got more mites here. This is Platynothus peltifer, or something like that. It's one of the, again, one of the orobatids, very common ones in leaf litter and moss quite often as well. That's this rather lovely ornament of ridges over the top of them. So we'll definitely be able to identify that with no trouble at all. And that's a different sort of mite here. This is a mesostigmaton. That will be a female from the underside, which makes it one of the Parasitidae, probably a Pergamasus or something like that. Again, it's possible to ID those, but you need some quite big keys unless you know them off by heart, which I certainly don't. But that's certainly doable. And there's quite a lot of other things in here. A tiny little mite. This is so these springtails are quite small. I mean, they're probably about 1 millimetre maximum, and then you get these little opioid mites here. Which one's that? That one we're going to need a closer look at. It's quite hairy, actually. Interesting. That could be a Disorhina, maybe. But there are a large number of species which are very similar and need quite subtle details to tease them apart. But I'll have a good look at that one as well. Lots of others you can start to make out. Oh, what's that? That one's in side view. I'll just turn it over using the standard tool, which is a single hairbrush. This is a single bristle, and I need to hopefully do this. It's quite difficult when it's on this scale to actually manipulate things, but there we go. That's my bristle. And it's right next to the edge of the dish, so I'm going to have to move it around a bit so I can actually get an angle for it. Looks like Orobatula tibialis. very common little Orobatid mite, but we'll certainly be able to identify that as well. You're starting to get an idea of the diversity of things in here. That's a totally different springtail here. Let me turn the brightness down on that. This is one of the poduromorphs. It looks like it's basically all of the internal tissue is decayed. This is just the exterior, which is interesting. We can, again, should be able to do something like that. Quite often, what we want in order to see the fine details, which can be extremely tricky, is to actually clear the specimens with bleach. So what you end up with then is just the cuticle without lots of dark organic material getting in the way, and that's the only way to see some of the features. So that looks like it's been ready cleared, which does happen sometimes if it sort of dries out on the edge, for example. And oh, we have a fly larva, some sort of crane fly? Maybe? Not totally sure. That may be a little crane fly. There's a lot of tiny species of crane fly as well. There's another... Ooh, what's that? Yeah, some sort of fly larva again. Not totally sure. But you can see there's lots of these little blue springtails, which we will definitely be having a look at, because I've had a look at some already from the first couple of days of the sample running, and they were interesting, shall we say. But you're starting to get the idea. There are quite a lot of springtails, not so many mites. Still a fair number. A few quite big springtails. This is one of the Tomoserids, and we'll be able to identify that one as well by checking things like the exact morphology of the end of this springing organ. They're called springtails because they can jump, most of them, with this thing here. And at the end of this, it's forked. There's two parts of it. At the very end of it, there's that bit called the mucro, which has some extremely tiny spines on it and other structures, which you can't quite make out with this microscope. But we'll be able to use that to distinguish which species it is in case there's any doubt. And yeah, you get the idea. So we can swirl this around a bit just to concentrate things. In the centre. And then what we'll do is pick out all the animals aside from the mud using a little pipette, put them back into one of these vials, and then we have a nice clean samples that you can go through and start to identify everything in. Yes, everything. That's the idea. Sometimes there are ones that I freely admit I cannot identify because I don't have the keys. Quite often there are no keys for the UK species. I don't have the access to the papers, which might be in some language I don't speak. They might well not require morphological detail that I cannot get with my microscopes. That is often the way. I've sometimes tried keying out things like the Scutacaridae, which you always get one or two little tiny little round things with big bristly legs sticking out the end, called Scutacarids, and you always get some, and so far I've not been able to get the resolution of the detail that I need to be able to work through the keys. It's going to be possible, I think, but I might need a slightly better microscope or a slightly better technique, because I am not one of the world experts in this stuff at all. I'm sort of a tier or two below that, probably 3 or 4. But I can use what I've got here to identify as much as possible. Just put the lid back on so the alcohol doesn't evaporate. One of those things that's quite horrifying to find that you've forgotten to put the lid back on. Come back a few hours later and everything's dried up. Luckily, that hasn't happened for years, I'm glad to say. Honest. Right. So, I say, this is for the first sort. This is where you pick things out and get a vague idea of what it is you're looking at. If you want to do the next stage, you're going to need a different microscope. And of course, you only get to see this one normally. But there are others. And over here, we've got the one that we're going to need for this. If you've ever wondered how much effort it takes to identify a springtail, there are some common species you can see at a glance. You know, from a metre away you can see, aha, Orchicella cincta. But mostly it takes a bit more effort, and there is a published key to the springtails of the UK. And this is it. It's by the sadly late Steve Hopkin, who did an incredible job of compiling all the information that was known and putting it into a format that allows anyone with dedication and the right equipment to be able to identify almost any springtail you're likely to find in the UK. So there have been additions to the list since then, and there are some changes to our understanding and identification of them. But this is the standard sort of diagrams that you have in here, and you're looking at minute little details, the shapes of the claws and so on. But it is one that allows you to start at the beginning, work through, and eventually you come to what should be the correct species. At least that's the idea. And usually it works remarkably well. And it takes some practice to be able to use it efficiently. I now know that I can go straight to number whatever that is. If I've got something of a particular group, I don't have to worry about going through the whole key. And most of the time you can actually work it out fairly well. So let's have a little look at some of the details that we need for some of these. So pop this in here. I will just point out that I've actually got two light sources here. There's one, a built-in one, so you're looking at transmitted light, but I also have this huge, great swan neck 20-watt light source, which is very good for tiny fossils, which you can apply from the top. And that allows you to see reflected light, which can be extremely handy because it allows you to see the colour. And here it is. It's a rather lovely sort of purple-blue colour. It's in side view, obviously, here. And you can see this is in the reflected light, and you can see the colour quite clearly. Usually we have the transmitted light and the colour gets washed out. Quite often we'll just have the one light source rather than the other. It can be useful to have both. But if we go purely to the transmitted light, we have this instead, which gives us a lot more resolution of details. At least we'll be able to zoom in now and find a lot more. So change the lens over, and we have to refocus it, and now we've got the legs. This is sort of intermediate magnification now, but it's much better than we could see with the other microscope already. And you can see the claws on the end of the legs. Let's have a closer look at them. And that is one of its little feet. So what you can see here is the claw is the upper part on the right, and then pointing straight down is an empodium. It's a separate sort of adjustable bit that you get in some springtails but not others. And the details of the shape of that and of the bristles coming off the sides of the legs, off the side of the leg next to the claw, can also be very important. In some cases you have tiny little blobs on the end of the bristles, what we call clavate setae. And you can see them quite clearly with this, even though it's pretty tiny. The light sort of bends around them. But there's nothing of that going on there. But you can see the arrangement of the bristles, and the form of those can be quite important for identification. Here we've got one of the legs in the other angle, which means we're seeing bristles going out a different plane of section. Okay, let's have a look elsewhere. Right, let's aim for the furca now, this jumping organ, and that mucro, the tiny little bit at the end, which I pointed out in the other microscope. You can see we have the two branches of it, and they have this little spined mucro, or toothed mucro at the end. And one of the critical things is the number and arrangement of those little spines. And it's actually really difficult in this case, because as you see the different side of it, on the one hand you see 3, like on this one, you focus through it, you can make out there's 3. Go to the other one, it only looks like there's 2. And this can be because of the angle of superposition of different spines. And it turns out that there are indeed 3 spines, but 2 of them are virtually overlapping. Okay, some other features you need to look at. These bristles on its back, are those serrated? And do they have little sensory ones with tiny little filaments coming off them? These are not serrated at all. They're perfectly smooth bristles. But what we could really do with is Trichobothria, see if they're present, which are very straight bristles with tiny little feathery filaments coming off the side of them. And we're not seeing that here either. So there we go. We've got multiple features here that are useful for the key. Now, there's an air bubble that's trapped there. If you were to look at this if you were to work through the key for this, you have to go from one question to the next, answering details about particular features, and eventually it takes you to the right answer. It's a dichotomous key, so has it got 3 bristles on this bit of the leg or 2, and so on. One of the critical things is whether some of these segments on the abdomen are actually fused together, and this is actually quite difficult to tell, but we're looking here for whether abdominal segments 5 and 6 are fused. And sometimes they can be sort of partly fused or appear to be, but in this case it does seem as though they are not. There is definitely a distinction between abdominal segment 5 and 6, which is one of the critical branches in the key that takes us to various different genera. So that's good to see. Another feature that's quite critical usually is the number of ocelli, the individual lenses in the eye. And this can be really hard to spot. You have to get the light sort of bouncing off it at the right angle in order to see them. Quite often they're on a sort of dark pigmented patch as well, which makes it even harder. But here you can see there's a series of 4, a little square of them at the lower left side, focusing back and forth through those. And then there is another line of 3 curving up from the top right corner of those 4, and then there's a tiny little one between that line of 3 and the other 4. And that gives us 8 lenses, which is exactly what you'd expect for most of these. It's only really interesting if it's a different number. And quite a few springtails are blind as well. They tend to be white ones. Now we can have another look. This is a different specimen, so it's worth checking all the details. And again, we've got lovely detail of the claws, and there are no clavate setae on those legs. So definitely no clavate setae. That's a good thing. Again, check the furca, since this is a different specimen again, and we have an identical sort of pattern here. Very elongate, and you can see very clearly it seems to have just the 2 spines. And we know that these do actually have 3, but there are 2 which are virtually superimposed on top of each other when you're looking at them from the side, so you only see it as 2. It's actually quite an elongate structure though, this, compared with most of the ones that you would see in the key. This is a bit more stretched out than I'd expect to see. But still, that's a really nice example. If we move along the furca, then you go into the dens, the middle part. You can see the setae along the edge of it, and the arrangement of those can be important. And also the arrangement of setae on the manubrium, which is the basal part, where it's still one piece before it splits into 2. And the arrangement of setae at the end of the manubrium is actually a critical feature in these keys. It's not the easiest thing to see, because quite often you need to have the furca lying flat rather than in side view, and you have to look at it from the right side. So it's a fiddly one to try to actually understand properly. And to be able to answer the key points quite often is just very hard to do. So this is the manubrium, but it's from the ventral side. So we're looking at the wrong surface to really see these setae very clearly. And I'm just hoping that we'll be able to see through it with the transmitted light to see some of the features that we want. Because I've come down to a key point where the critical thing appears to be, or one of the critical things, a couple of spines that occurred on a plate just between the manubrium and these two branches of the dents. So we're looking at a fairly subtle feature here, and unfortunately we can't see it from the ventral side. So we're going to have to actually get a specimen lined up the other way up, and then we'll be able to actually catch these details and hopefully also look at the arrangement of the bristles in this area, because that is also critical. So here we are. This is the right way up, and we're looking for tiny structures in that gap, superimposed on the soft tissue, just set back in slightly, just from the end of the gap between the two parts of the dens. So, what can we see? Not a huge amount at first glance. You actually have to spend quite a long time fiddling with this, and then you finally can see that it's got a pair of spines on each side rather than just one. And this is within the genus Isotoma now, assuming everything has gone right to get to this point in the key. That means that it's not Isotoma viridis, which is hardly surprising because that's green, and it narrows it down to Isotoma anglicana or Isotoma cerulea. And now we can see these bristle arrangements here, and that doesn't seem to match either of them. Typical. Now, anyone who's used to using these sort of keys will know that it doesn't always work out. Sometimes you just go wrong. I mean, you might have to go through 20 or 30 key points, some of which can be quite subtle. Are there 7 or 8 ocelli there? But usually, with a bit of practice, you start to go wrong less and less often, and I've had quite a lot of practice at this key now. So when you end up with an option of 2 different species, after having gone over and over all of the key points to get to that point and being quite happy that you haven't gone obviously wrong anywhere else, you start getting a bit intrigued. So you start looking at all of the confirmatory characters that you can find, see if you can find any other differences or similarities. Now, both of these 2 species that the key comes out at are up to 4 millimeters long. They can be bluish, but not strictly blue, apparently, whereas these are really quite blue. And they're up to about 1 millimeter long, maybe 1.1, 1.2. So what we've got here, and we've got a lot of them, is either a juvenile population with nothing approaching full grown, or it's something which is much smaller and much bluer. So you need to look at details of the other characters that you can see. And things like the form of the mucro on the end of the furca, that little toothed bit. And what we've got here is actually much more elongated than the typical mucro that you get in this genus, in Isotoma. So, does that mean something significant? It's difficult to be sure because it's not illustrated for the particular species, but they should all be very similar within the genus. And if you go back a few points and think, well, could it be an Isotomurus? Could there have been Trichobothia that have been rubbed off on the back or something like that? We just don't happen to see any. And if you go down that route, well, firstly, there's nothing that matches the colour of it. And secondly, the mucro is again different. It's got an extra tooth on the end of it. So we can kind of rule out that. You start going round and round trying to find ways that you've gone wrong. And eventually, sometimes, you fail. You start wondering, well, is it something that's not in here? Is it something that's new to the UK, but is known from European keys, for example? So then you have to start finding European or global keys and try to work out if there's anything there that isn't known in the UK yet. Or could it be that you've actually got a new species? How likely is that? I mean, in paleontology, we find new species all the time. You know, kind of disappointed if we don't find any. But in biology, zoology, it's actually much harder. If you're studying things in the Amazon or Congo Basin or something, then yeah, absolutely, you will find new species. Deep sea creatures, no problem at all. But somewhere like the UK, which has been studied so intensively for centuries, to find something that has been overlooked not only here but across Europe as well, assuming it lives anywhere else outside the UK, that takes some doing. I certainly have added a few species to the UK list, things that are known from Europe, and it does happen. Both leafhoppers and mites, several have done for that, but it's unusual. I mean, you get quite excited when that happens because it's not normal. There are something like 300 species of springtails in the UK, and that's a reasonable number, but every now and then there'll be another new one. Now, there are a couple of unnamed species in the UK, probably 3 or 4 actually, but of springtails, but they have come from somewhere like New Zealand. They've never been found in the natural habitat, but they have been naturalised in the UK and now they're popping up everywhere. And they're very well known, it's just nobody's got round to naming them. But to find something in a little boggy field in the middle of Wales which doesn't seem to match up with anything that is already known, that's harder to justify as a conclusion, and I am not going there yet. But at the moment, I've drawn a blank here. And it may well be that there's someone amongst you who is actually a European springtail expert and can say exactly what that is straight away. And it's possible still that I have missed something obvious. I don't claim to be someone with totally encyclopedic knowledge of these, but I know my way around them. And this is something I don't think I've come across before. And I don't think I can make it fit into this one. So it's a really interesting thing, and with springtails, most of them key out nicely, but every now and then you get something really intriguing. So one thing I'm going to be doing is checking against all the other samples that are taken, see if we find the same thing in any of the other ones. I've already gone through the entirety of a second sample from about 50 meters away from where this one came from, but there was nothing in there that showed anything like it. There was one that was superficially similar. It was blue, the same size, same general appearance, but that was a pseudoisotoma. A sensibilis actually fits perfectly with what you would expect for that. So it is not that. There are numerous things like the clavate CT. It's got serrated CT on its back that are not present here. So this is something quite different, despite the fact that it basically looks identical. All right, let's show you a mite or two as well, just because they're fun. Let's have a go at one of those little tiny ones that are called an opioid and see if we can work out what that is, because it should be pretty straightforward, hopefully. Now, for springtails, there is a nice handy key to the UK species. For mites, it's a little bit different. So we have here a key to the German orobatid mites. And this is fairly substantial, as you can see, and it includes more than there is in the UK, but it does include, hopefully, everything more or less that we do have in the UK. There are a few exceptions, certainly things that occur here, but not in Germany. But in general, this will get you most of the way there, and the only downside is that it is in German, which is not exactly my best language at all. Not that I've got many good languages, but it's a very nice book, really good illustrations, but you need to go through the key because there's a lot of subtlety. Now, I mentioned with springtails, there's about 300 species in the UK. With mites, there are supposedly around 2,500. But they are much less well studied. So quite often I find mites that are new to the UK or which are just difficult to identify. Sometimes the level of detail in the features that's required is beyond what this poor little microscope can do, and I just have to leave it as sometimes even a family. I can't even get to genus on them. But we can go quite a long way. And these little orobatids, they've got a good chance of giving us an answer. So let's have a look. So here it is. This thing is about 0.3 to 0.4 millimeters long. It's rather cute. You can try the different lighting effects. We need some higher magnification for this, so let's have a little look. Firstly with the reflected light mainly, and you can start to see the basic form of it and some of the details. It's got these little sort of bobbly legs, a big abdominal part, the notogaster, and then this front part which has some interesting structures on it. You have to go in even closer to see the detail of those, because we're going to need some nice detail on this. These little opioids, there are about 50 odd species in the UK that might be, so we're going to need some subtlety. And let's have a look at this, the prodorsum, this front part, and you can see these ridges on here, sort of almost hourglass shaped things. And also ridges going backwards onto the abdominal part, the notogaster again. So you have to focus up and down through it in order to see much of the detail that's going on. You need to change the lighting effects. Sometimes transmitted works better, sometimes reflected. A combination of the two can do wonders. And by going up and down through it, you can start to work out how those ridges actually work. Now go fully into transmitted light, and there's a bit sticking up here, which disappears when you focus down again, but this is a thing called the sensillus. It's a tiny little, well, it's a large bristle, still tiny though, which it uses as a sort of sensory device, and these often have very complex shapes and interesting structures which you can use to identify them at genus and species level. So it looks like this is going to be an Opiella species, and probably a thing called Opiella nova. Now we have a side view to see the sensillus properly, and now we can see the shape of it. It's this long spindle with a little pointed tip coming out the end. So yeah, that kind of gives us the answer that we're expecting. It's a very common species, in this case, Opiella nova. Nice. There are so many more things in these samples that, well, it would take forever, but I will show you a little gallery of some of them at the end, because the sample that I showed you collecting at the beginning, I have now finished, and I'll give you a rundown of what was in there and how diverse this sample is, just to give you a flavour of it at the end. But what I want to get onto a little bit is what we can learn from looking at these types of assemblages about how we interpret the fossil record. I mean, to some extent, it's going to be a little bit difficult because we're looking at a terrestrial thing here, whereas most of the fossil record is marine. They're also tiny little things. So mites and springtails have, unsurprisingly, a very poor fossil record. Mostly they're in amber. So it's young rocks with resin in that came trickling down trees, and they was in the habitat where they were living. But they do both go back to the early Devonian or even the late Silurian, where we have some of the first terrestrial arthropods on land. The Viney Chert and Gilboa site in New York. I will be covering these because this is a really interesting story. I'll probably do this fairly soon, where I get into what we know about the first of these arthropod lineages to colonize the land, and the first land plant assemblages that went with them. So what we do know is that they can fossilize under extraordinary conditions. And some of these things are the right size to fossilize at Castle Bank. So, you know, we are keeping an eye open for anything that might look like a marine mite. Yeah, doesn't really seem likely, but there must be precursors there somewhere. So we will be looking out for that. But we can say quite a lot more about the nature of ecosystems, of diversity patterns. So one of these little samples of mud can have 50, 100 species in it, in a tiny area, of things that have a cuticle, so are not impossible to fossilize. Certainly these little orobatid mites, there are many, many species of those, and they are fairly resistant. You don't need truly exceptional preservation, not the softest anyway, in order to fossilize that. You get the right conditions, you'd expect to see them. But what would you see? Because we have here a typical hollow curve distribution pattern. We have a few species that are common, and then a long, long tail of things that are rarer. That's within one sample. You start comparing samples, and you find that there are a few species that occur in multiple samples. They're ubiquitous. And then in each sample, you have a substantial number of species that you don't find in the ones next to it, because they have either very specific habitat requirements, or they have very low population densities such that you're only likely to turn them up every now and then. Or they're just really difficult to identify, because we're dealing with modern things here when you go down to these tiny levels of detail, and that's what you need to identify them. With fossils, you're not going to get that resolution, not to the same extent. So you always have to bear in mind that what we call one species as a fossil is likely to be quite a lot of species biologically. Quite likely, anyway. And then you have the things that have been overlooked for whatever reason and are genuinely rare in the modern. So in this sample that I've done completely, there were 2 species that I have added to the list for the UK, personally, because they'd been found before in some cases, in one case at least, but overlooked. It hadn't been recognised what it was because it's quite similar to another one. The other one is a tiny little fluffy thing which I've now found in Malvern, about 50 miles away, and now here as well. So it seems to be a thing which is not that uncommon in western parts of Britain. But maybe the western parts of Britain have not been sampled so well in the past. This is an area with relatively low population of acarologists. So we normally find that anything that I've not collected before that turns up in mid-Wales, for mites anyway, is new to mid-Wales because nobody else has ever looked either. And most samples, you're not going to get those in, but every now and then they turn up. When you're looking at the fossil record, you have a lump of rock, or quite a large amount of rock, but it represents one precise location on the seafloor. And it's one precise habitat. Things can get washed around in the sea, they can be concentrated in some areas, but they can also be concentrated in the place you're not looking at. We may have lots of things here that might have been fossilized, but have been washed away, they've been winnowed, they've been deposited somewhere else, they've been eroded. There are so many filters on the fossil record that prevent us from seeing things. And so what we get generally is a selection of the common species. When I say common, I mean the ubiquitous ones, the things that we are getting in every sample, the things that are abundant in most of the samples, that have a high chance of making it into the fossil record. We also have rare fossils, of course, but those rare ones might be the equivalent of the ones that are ubiquitous, but in lower numbers. So maybe you only get 3 or 4 of them in each sample, rather than 20. As for the things that we find as 1s and 2s in just 1 or 2 samples, they're never going to make it into the fossil record. For the most part, most of life is not going to be preserved, which is slightly depressing. But the problem is that I think often we think of rare fossils in the fossil record, so things that have turned up just 3 or 4 times, but if you go to the right place, you have a chance of finding it. So uncommon trilobites and so on. We think of those as being biologically rare. They're not. If you think of something in the UK which is biologically rare, take the Cornish shieldbug, which we happened to go and find when we were on our honeymoon, because that was, what you do when you have a nice sort of holiday to go on. And the Cornish shieldbug exists in the UK at one site, Sennan Cove in Cornwall. It lives in one particular habitat at that site. It was once found about a century ago in South Wales, but that population seems to have gone. That's what we mean by a rare species in entomology, something which is highly unlikely to be found anywhere because it doesn't exist there at all. And if you go looking at rock pools, almost everything that you find as a casual rock pooler, you will find at pretty much every site which has the same sort of habitat. It's very unusual to find something that you can identify that you've never seen before and that you don't find again when you start looking for it, which is only restricted to a few places. That's what we mean by rare. And it's important to realize this, that when we're seeing an ecosystem of maybe even 10 different trilobites in one section or one formation, and we get quite excited by the diversity of them, that's probably not the full diversity. That's probably just sampling the tall end of the hollow curve, and there would have been lots of others that were in low populations, which most of the individuals got broken down, the shells were smashed, they're not in recognizable form anymore, and we just don't see them at all. It's a really interesting area, and I want to explore the impact of rarity on the fossil record a lot more in future videos. So this is kind of leading into it a little bit, but there is so much more that can be said about it, and I think I probably will. So, yeah. So this is one of these areas where modern zoology and the fossil record tie in perfectly, that you need that understanding of how things work in the modern in order to interpret what we see as paleontologists. And it can't be overstated that if you're a paleontologist, you really ought to spend some time looking at the modern world as well, because there's a lot more there to it that we don't normally pay attention to and don't really understand. You can be misled quite easily by trying to interpret a set of fossils or bedding plane assemblages at face value. Anyway, that's enough science for the moment. Enjoy some pretty pictures and some harping.